Hello and welcome back to another video where today we're going to be discussing DNA. So this is going to be part of a two-part series on DNA and RNA because it can be quite a complex topic to get your head around. So we're just going to take it slowly, and today we're just going to discuss DNA, its functions and its structure as well, before in the next video going on to DNA replication. Okay, so let's start looking at the basic structure of a nucleotide. A nucleotide consists of three main components: a pentose sugar, which is a 5-carbon sugar, a phosphate group, and a nitrogenous base. These bases can be adenine, A, thymine, T, cytosine, C, or guanine, G. These nucleotides are the building blocks of DNA. DNA is made up of two strands of nucleotides. These strands run in opposite directions, meaning they are anti-parallel, and they are joined together by hydrogen bonds between the nitrogenous bases. This bonding between the strands gives DNA its stability, which is crucial for storing genetic information over time without degrading. It's this structure that enables DNA to act as a blueprint for life. Now, let's talk about base pairing. The nitrogenous bases are very specific in how they pair. Adenine always pairs with thymine, and cytosine always pairs with guanine. This is called complementary base pairing, and it's a feature that ensures the DNA strands match up correctly during replication. This precision is vital because even a small error in base pairing can lead to mutations. The nucleotides are connected by phosphodiester bonds, which form between the phosphate group of one nucleotide and the sugar of the next. These bonds link the nucleotides into long chains called polynucleotides. Two of these long polynucleotide chains come together and twist around each other, forming the double helix structure that DNA is famous for. This double helix is not just for appearance, it plays an important role in the molecule's stability and function, as we'll discuss later. An important feature of this structure is the sugar-phosphate backbone. The backbone, made of alternating pentose sugars and phosphate groups, provides structural support. It helps to protect the chemically reactive bases inside the helix, keeping the genetic information safe and less prone to damage. Okay, so let's summarize this slide. The structure of DNA is perfectly suited to its function. The double helix, the complementary base pairing, and the strong phosphodiester bonds ensure the DNA can store, replicate, and pass on genetic information with incredible accuracy and stability. This allows cells to function, divide, and produce proteins necessary for life. Okay, so now we've taken some time to go over and discuss and familiarize ourselves with the structure of DNA. Let's take a look at the function. So DNA is essential for passing on genetic information. It's responsible for the transmission of genetic material from cell to cell during mitosis, as well as from one generation to the next during reproduction. This ability to reliably transfer genetic information ensures that characteristics are inherited and allows for the continuity of life. Now let's explore how DNA is adapted to its function. One of the key features of DNA is its stability. DNA is incredibly stable, which means it rarely mutates. This stability is vital because mutations in the DNA sequence could lead to errors in protein production, potentially causing diseases. This stability comes from its strong phosphodiester bonds in the sugar-phosphate backbone, which keeps the structure intact over time. The two strands of DNA are held together by hydrogen bonds between complementary base pairs. These bonds are relatively weak compared to covalent bonds, which is important because it allows the two strands to easily separate during processes like DNA replication and transcription, as we'll discuss later. This separation is necessary for cells to copy their DNA and pass it on to new cells or produce mRNA for protein synthesis. DNA is also a very large molecule, which enables it to carry an immense amount of genetic information. This information is stored in the form of sequences of bases, adenine, thymine, cytosine, and guanine, which provide the instructions for building proteins. The length and complexity of DNA allows it to encode the vast array of proteins required for life's functions. One of the ways DNA protects its genetic information is through its sugar phosphate backbone. The backbone shields the more chemically reactive nitrogenous bases that are inside the double helix, protecting them from damage and degradation. This ensures that the genetic code remains intact, even in challenging environments. Another important concept to understand is the directionality of DNA strands. This can be quite difficult to wrap your head around. DNA strands run in opposite directions, described as 5' to 3' and 3' to 5'. This anti-parallel arrangement is crucial for DNA replication. DNA polymerase, the enzyme responsible for adding nucleotides during replication, can only add new nucleotides to the 3' end of the growing strand. This means that the DNA is always synthesized in the 5' to 3' direction, which is a key feature of DNA's replication process. So to summarize this slide, DNA is perfectly adapted to its role in storing and transmitting genetic information. Its stability, size, ability to separate its strands for replication, and its specific directionality all work together to ensure that the genetic code is accurately passed from one cell or organism to the next, allowing life to continue and evolve. So that concludes this first video on DNA. We discussed the function and structure of DNA. Make sure to check out the next video on DNA replication. With that being said, thank you very much for watching. Make sure to like, comment and subscribe, and also feel free to check out the multitude of other videos on this channel on A-level biology. Thank you.